Welcome to the second session of metabolism of amino acid. And in this session, we'll be discussing about the catabolism of amino acids, the general aspects of amino acid metabolism, and we'll be discussing in detail about transamination and the mechanism of transamination. In the forthcoming video, in my next video, I'll be explaining the continuation of this amino acid metabolism, that is deamination, oxidative deamination, non-oxidative deamination, decarboxylation, and urea cycle. So by the end of this session, you will be able to understand the metabolism of amino acid in brief, the general aspects of amino acids, what is transamination, examples of transamination, silent features of transamination, and mechanism of transamination. So coming to the catabolism of amino acids, the amino acids undergo certain common reactions like transamination, which is followed by deamination for the liberation of ammonia. The amino group of the amino acids is utilized for the formation of urea, which is an excretory end product of protein metabolism. Later we will be studying also about the decarboxylation reactions of amino acids. So the amino acid is first converted to keto acids by transamination, which meets one or more of the facts. So these keto acids, or like the nitrogen in the keto acids, are utilized for the generation of energy. It is used for the synthesis of glucose. That means it contributes to the Krebs cycle. It is diverted for the formation of ketone bodies or fate of ketone bodies, the breakdown of ketone bodies. It also is involved in the production of non-essential amino acids. The general aspects of amino acid metabolism, or amino acid metabolism, or catabolism of amino acid, or like oxidation of amino acid, fate of nitrogen, all these terms give the same meaning. So coming to these fate of nitrogen, or catabolism of amino acid, or metabolism of amino acid, or oxidation of amino acid, mainly three steps are involved. The first one is transamination, second one is deamination, the third one is decarboxylation. So the names over here itself suggest us what does it mean. Transamination transfers the amino group from one amino acid to another amino acid. Deamination is the removal of an amino group from the amino acid, and decarboxylation is removal of carboxylic acid group. As mentioned in the previous slide, transamination is a process or the transfer of amino group from an amino acid to a keto acid. This is what we call it as transamination. So the transfer of amino group from an amino acid to a keto acid is called as transamination. So over here we have amino acid 1 and a keto acid 1. So from this amino acid 1, the amino acid group or the amino group is being transferred to the keto acid. So when it is being transferred to the keto acid, so this keto group is being transferred to the amino acid 1. So in this process we can see over here, so this keto acid is converted to a different amino acid, and this amino acid over here is being converted into a keto acid. So this process involves the interconversion of a pair of amino acids and a pair of keto acids. This is catalyzed by a group of enzymes called as transaminases. Coming to the silent features of transamination, all transaminases require pyridoxal phosphate, a coenzyme derivative of vitamin B6. Specific transaminases exist for each pair of amino and keto acids. For instance, aspartate transaminase which transfers amino group from aspartate, alanine transaminase, which transfers amino group from alanine, and so on. There is no free amino group which is liberated out. Only there is transfer of amino group which occurs over here in transaminases. Transamination is a reversible reaction, and transamination is very important for the redistribution of amino group and production of non-essential amino acids. The transamination diverts the excess of amino acids towards energy generation. The amino acids undergo transamination to finally concentrate the nitrogen in glutamate. So ultimately, in transamination, the aim of transamination or the goal is to form glutamate ions. So these glutamate ions undergo oxidative deamination to a significant extent to liberate free ammonia for the synthesis of urea. All the amino acids except lysine, threonine, proline, hydroxyproline participate in transamination reactions. The transamination is not restricted to alpha amino groups only, and the serum transamination are very important for diagnostic purposes. So these are the salient features of transamination. Here is an example for transamination. We are using the amino acid alanine and a keto group or alpha ketoglutarate in the presence of alanine transaminase and pyridoxal phosphate. The amino group of the alanine is being transferred to the keto group to form a new amino acid. Coming to the mechanism of transamination, the mechanism of transamination can be explained in two different stages. The first stage or the first step includes the transfer of the amino group to pyridoxal phosphate, the coenzyme pyridoxal phosphate, to form pyridoxal amine phosphate. The second step or the second stage includes the amino group of pyridoxal amine phosphate is and the enzyme with pyridoxal phosphate is being regenerated. So as I have mentioned, like the stage 1 includes the transfer of the amino group to the coenzyme pyridoxal phosphate to form pyridoxal amine phosphate. Coming to the reaction involved, so this I have taken an example of alanine. Alanine, when it is being treated with pyridoxal phosphate, so the amino group of alanine is being transferred to the pyridoxal phosphate to form an intermediate compound over here. So there is some rearrangement in the structure which takes place, and ultimately it forms pyridoxal amine phosphate over here. So over here you can see the formation of pyridoxal amine phosphate, and even the pyridoxal amine phosphate which is being formed over here is an intermediate molecule. The stage 2 involves the amino acid group pyridoxal amine phosphate is then transferred to a keto acid to produce a new amino acid, and the enzyme with pyridoxal phosphate is being regenerated back. So pyridoxal amine phosphate, which we have synthesized in the previous slide over here, will react with the keto acid. Over here the amino acid of is being transferred from the pyridoxal amine phosphate to the keto acid or the keto group over here to form a new amino acid, and we can see over here that pyridoxal phosphate coenzyme is being regenerated back. So by the end of this session, I hope you are able to understand the catabolism of amino acids, the general aspects of amino acid metabolism, what is transamination, the silent features of transamination, what is the mechanism of reaction in transamination. So in my next video, I would be explaining you what is deamination, what is oxidative deamination, what is non-oxidative deamination, what is decarboxylation, and we'll be discussing about urea cycle in my next video. Thank you very much. Namaste.